Hello, so today we'll talk about hemoglobin. Hemoglobin is a very important molecule found in red blood cells that give it the ability to transport oxygen that we breathe in the lungs to the tissues that need it for metabolic processes to break down the macromolecules that will give energy for the cells to do the things it needs to do. So to jump right in, we'll talk about the structure of hemoglobin. Hemoglobin, found in red blood cells, is comprised of alpha and beta globin chains. So these are one alpha, one beta dimerized, and there are two dimers. Now, each of those globin chains has a heme group represented here, and each heme group has a ferrous Fe2 plus ion, and each ion has an oxygen molecule. So overall, what we have is that each hemoglobin can bind up to 4 oxygen molecules. And there are two main states that we refer to hemoglobin in being. It is either in the deoxyhemoglobin state, most notably referred to as the T state. And this is a state in which there is a lack of oxygen molecules, in which there is a lower affinity to oxygen, so it's releasing oxygen. Oxyhemoglobin, on the other hand, is when it's saturated with oxygen, and it is in the R state. So, how does oxygen bind to heme? Now, to understand how that works, let's take a quick look at the heme group structure. So, it's a protoporphyrin ring with an ferric Fe2 plus iron in the center. Now, if you look at it from the side, we see that it can be portrayed in two different ways: the deoxyhemoglobin and the oxyhemoglobin heme group. Two different structures, and this is the case in which the deoxyhemoglobin, when it binds to oxygen, it will become smaller, and it'll change structure so that it moves into the plane, along the plane of the porphyrin, and as a result, this changes the structure of hemoglobin. How does it do that? Well, if we take a look at the histidine ring, this histidine ring is connected to the subunit of the globin chain. And what it does is it pulls the alpha helix within that subunit, and it transitions it from the T state into the R state. And as this transition occurs, what happens is that because of cooperative behavior, other subunits within the hemoglobin start to transition from the T state to the R state as well. And we get this change from deoxyhemoglobin to an oxyhemoglobin. And as a result of cooperative behavior, it becomes easier for hemoglobin to bind to oxygen, the more oxygen molecules it has. And one thing I'd like to point out, though, is that it's very important that iron is ferrous. If it is ferric, which is when it's oxidized, it's unable to bind oxygen, and that's a problem. So, moving on, what are the factors that result in a difference between oxygen in the lungs versus oxygen in the capillaries or the tissues where it needs to be released by heme? Well, what happens is there are a few factors that contribute to this representation of an oxygen dissociation curve. So this graph illustrates that as it shifts to the right, certain factors contribute to this shift to the right, which represents a decreased affinity for oxygen. So this would be, for example, represented by the T state, when it's needing to release oxygen to the tissues. And those factors that contribute to that would be an increase in carbon dioxide, which is a result of the metabolic processes in the tissues that release the CO2, an increase in protons, which lower the pH, as well as 2,3-ppg levels are increasing, resulting in this shift, and increased temperature, which we won't go into too much detail. So those are the factors contributing to the decreased affinity for oxygen. Now on the other side of the spectrum, the shift to the left on this curve involves an increased affinity for oxygen, which we can represent as being the R state or the oxyhemoglobin. And this is just the opposite of the factors we had just gone over. So if we take a look at 2,3-ppg, it has an effect of releasing oxygen to the tissues. How is it able to do that? Well, if we look at the structure of 2,3-ppg, it has negative charges on particular groups that can bind within the center of the hemoglobin tetramer, and it will form ionic bonds between some of the globin chains that have positive charges to it. As a result of these ionic bonds, and this is ionic bonds within the T state, as a result of that, the T state becomes more stable, and if the T state is more stable, it is the preferred state. If it's the preferred state, then it will have a lower affinity for oxygen, and oxygen will be released to the tissues. So as a result, 2,3-ppg is important for releasing oxygen because it favors the T state. Now, oxygen, in reference to the pH, we'll refer to as being part of the Bohr effect. When we consider pH, there are varying degrees of pH as we look at the lungs compared to the capillaries in the tissues. There will be a higher pH, around like 7.4, and a lower pH compared to the tissues, where the tissues would have something more like a 7.2. Seems like a small difference, but it creates a drastic effect where there's release of oxygen at the tissues. So what is going on here is that the T state there are ionic bonds between the beta 146 histidine and the beta 94 aspartate. So these bonds are possible as a result of the positive charge on the histidine and the negative charge of the aspartate. If the pH were to go up, that histidine would become deprotonated, and as a result cannot interact with aspartate. As a result, this means that the decreased ionic bonding will cause the T state to be less stable. If it's less stable, it is not favored, in which case the R state would be favored, which means that it would try to hold on to oxygen. However, if the pH was decreased, the histidine would be positively charged, form bonds with the aspartate, and thus the T state would be more stable. And if it's more stable, it's preferred. And if it's preferred, then oxygen gets released to the tissues. So that is one major reason as to why pH has an effect of releasing oxygen, and that is because it favors the T state with the ionic bond interactions. Now, if we take a look at CO2, CO2 has an effect that we will refer to as the Bohr effect as well. So two things that play a factor in releasing oxygen is the formation of carbamate groups and its effect on the pH within the blood. So as CO2 is being produced by the metabolic processes within cells, that CO2 can bind to the terminal groups of T-state hemoglobins. If it binds to it, we have the formation of what is referred to as a carbamate group. And those carbamate groups, being that they're negatively charged, can then interact with the globin chains, the positively charged portions of the globin chains, to form ionic bonds. And by doing so, it becomes more stable. So by forming these carbamate groups, we get more stable T states, which means T states get favored, and then the there is an increased release of oxygen to those tissues. Now, if we also take a look at the effect of CO2 and pH, what we see happening is that CO2 can actually dissolve into the water. It can do so spontaneously or with the help of carbonic anhydrase. So what happens is that carbon dioxide will dissolve into the water as carbonic acid, and if it does so, then what it can do is ultimately release protons as it goes down into bicarbonate. So this is critical because, as we had just mentioned, when we look at protons, it lowers the pH, and the pH has a major effect on whether oxygen is being released or held onto. Here we see that with CO2 going up, there is going to be a release of oxygen because of that decrease in pH.